Thank you. So most of you have probably not heard of orbit discovery. There's a very good reason for that. We are brand new to the world. We were founded in November, so with probably maybe the latest company to enter the peptide space. We're a spin-out from University of Oxford, and we're based on we have funding from 3 different Oxford investment groups. It's all very local. And they're all a bit different. So Oxford Technology Management is a classic seed fund. Oxford Technology Innovation EIS fund is actually the fund behind Adaptimmune and Immunocore and some of those companies. And an EIS fund is a fund that enables individuals to invest very tax efficiently. And then Oxford Science and Innovation is our lead investor. They're actually a company, not a fund. They have a balance sheet of about £350 million, and the brief is to invest that on building very valuable companies in the Oxford area. And so we hope that we'll become one of those. And we're doing it with a focus on peptides. We're based in Oxford, and so we're in a very lively bioresearch space. And really what we're going to try and do is demonstrate that we can bridge the gap between what happens in academic labs where there's an enormous amount of disease area expertise and take drugs from there to the point where they're appropriate for commercial drug development. So this is the team. The colours change. We've all developed quite good tans. This is when I just came back from holiday. This is Graham Ogg. He's at the Weatherall Institute in Oxford, and it's in his lab where our technology was developed. And also at the Weatherall Institute is Terry Rabbits, who has also contributed to the development of this technology in the academic environment. And then our scientific advisory team. Andy Hamilton is a reasonably renowned peptide chemist with an enormous network. Jason Chin is in Cambridge. He's our expert in non-natural amino acids. And Mike Owen has been around in the pharma industry for a while, was at GSK for quite some time. Now he's on the board of Zealand, who apparently have an interest in peptides. And so we have different pieces of expertise advising us, and we're still building on this. I mean, there's a couple of key pieces of expertise that we need to bring in, but so far we feel that we're getting a very solid team. So our plan is somewhat phased. So we raised investment based on a discovery platform, which I'll explain. And so our first job is to move that platform from the academic lab and put it into a company environment and really validate it and demonstrate it, make it efficient, do a lot of process development, and really turn it into something that's more industrialized, like other discovery platforms. As part of that, we have a plan to engage pharma. So we are not thinking of pharma as our customer at this stage. So we are not broadly in the business of this thing's got a mind of its own. Broadly in the business of trying to set up broad collaborations in the style of, say, Peptide, for example. That's not our model, but we feel that working with pharma is going to be absolutely critical to ensure that our processes become robust and we behave like a real company. But then ultimately what we do is drug development. So our sort of medium-term goal is to become a drug development company. And in order to do this, we use our platform to identify hits, maybe early-stage leads, and then something to the point where we can bring in asset-specific investment to take programs forward. So the technology is a peptide display technology. It's based on a bead amplification, where we have a bead that has unique DNA attached to it. And in emulsion, we replicate those, and then we do in vitro transcription translation to end up with a bead that has peptide coated on the outside and the DNA labeling it inside. This thing's going automatically. It's very frustrating. And that bead links the phenotype to genotype. And in our library, we can have many millions or low billions of beads, and each one displays a different peptide. And we pass those peptides by our target, and then harvest either what sticks or what doesn't stick, depending on what we do. And we then use deep sequencing to identify what the peptides are that have bound. And that really starts an iterative process where we look at the peptide sequence, sometimes synthesize, typically generate more further libraries where we may lock in some pieces. And generally, we're trying to make things move from identifying a binder, making it more mature, reaching some kind of target affinity, then moving on, demonstrating function, demonstrating selectivity. And really, the endpoint for our technology is when we've demonstrated that the molecules that we've generated work in some sort of relevant disease model. So the proof of concept has been really driven by academic areas of interest and what material has been available. There's no really good rational reason for choosing any of these, but each one demonstrates different attributes of our base technology. So at the Weatherall Institute, where we're from, then there's a lot of expertise in HIV. So GP120 binding protein all the materials are available, the assays are available. In Terry Rabbitt's lab, there's expertise in RAS. So again, we had all the tools available. And actually, the real reason that this technology was developed is because in Graham's lab, there was an interest in deorphanizing T-cell populations, and none of the other methods for displaying peptides seemed to be able to support that. And so that was actually what triggered this technology. But that really demonstrates an ability to probe cell surface targets. So just to show some of the data. So for the GP120, we took a small library, 9 random amino acids, all natural, and we present those on a scaffold protein, so beta-2 microglobulin. And in a single round of selection, we're able to identify various peptides, some of which bound reasonably well. And then we transfer those into a cell-based assay where we're looking at inhibiting replication, inhibiting infection, and we've got the control T20 fusion here, and one of our molecules, albeit at a fairly high dose, was able to inhibit HIV replication at a similar level to fusion. So this came out of a single round of selection. And you may claim it was lucky, but it's fairly encouraging data. If we then go on to look at RAS, so RAS got a mention yesterday. Obviously, you'll know the problem with mutant RAS is that it leads to you get constitutive signaling, and so there's no control. And the real challenge with RAS would be to find a molecule that could bind to the mutant, but maybe not bind to the wild type. And block this constitutive signaling. So in Terry Rabbitt's lab, they worked out the crystal structure of RAS with a antibody domain, and the antibodies binding to this region here, which is the switch region, and basically using some in silico design, came up with a series of peptides that bind to that region and inhibit RAS. We thought it'd be interesting to see what happened with a randomized approach to the same thing. And so using our method, we again tested a random library, found some molecules that did bind to the mutant form of RAS. And interestingly, one of them, this one here, when we looked at the sequence, it was remarkably similar to two of the designed peptides. So again, another really good piece of luck, but all encouraging stuff. So we took that molecule along with the sort of rationally designed peptides and put them into a competition assay, and you can see that the orbit peptide here sort of falls in the middle. These are the two rationally designed ones. This is just another peptide in the series, and what that's doing is it's blocking the interaction of the RAS mutant with its ligand. And the final piece of sort of example is the cell surface target idea. So here we're looking for peptides that bind to T-cell receptors, and in this case, the bead structure is slightly different. So we have obviously the DNA that codes for the peptides here, and then beta-2 microglobulin as a scaffold that presents the randomized peptide on the outside, and then we recombine it with HLA heavy chain before the screen, and then incubate that bead library with our T cells. Instead of doing it in a capillary or on a surface here, we're doing it on a membrane in a filter well. Then we simply wash away everything that doesn't bind, take the beads that do bind, do the sequencing, and see what we get. And what we were able to show is that for T cells with known specificity, we've got 3 examples here, we preferentially pulled out the amino acid sequences that we were looking for. So that's fairly encouraging in terms of being able to probe a cell surface target and come up with some sort of specificity to the receptor rather than just to the cell itself. So just where we are, I say company evolution, we're right at the beginning. We're at conception stage for the minute, but what we're bringing together, obviously the expertise of the founders and expertise within the university. We've got some great investors. They're all different, all looking for different types of exit, but generally supportive of this phased concept, starting off with a platform and then turning into a drug company. And right now our focus is on the platform really getting it established and developing a subset of target type expertise. So there's no way we can do everything, and we need something that will bring us some focus on different types of target rather than different disease areas. And then as we get interesting molecules, then obviously they're our molecules, so we start with them. We raise asset-specific investment to take those molecules forward. And at this point, obviously, the target and disease area expertise becomes much more important. And these have much more obvious exits than this, which doesn't really have an obvious exit. And what we hope is that with the breadth of research that's going on in the university and the number of people who've got a lot of disease area expertise, are spending a lot of time validating targets and demonstrating possibilities for translation, that we'll be able to, where it seems appropriate to drug those with a peptide, then we'll be able to work with those academic groups, get their expertise, but actually develop early stage molecules that the pharmaceutical industry might be more interested in than something that came straight out of the university. So our initial target is to do one of them, and then over time we might do number 2, we might do number 3, really have the platform spawn these projects, and really just see where it goes. So that's orbit discovery. Thank you.
Thank you, Alex. It's great to see a company right at the embryonic stage. We have time for questions. I do have one. Where do you think your platform has its best application?